Hello and welcome to Oro-Oto-Cerebral Pathway. In embryology series, it's time to go systemic. In systemic embryology, we are excited to start with the cardiovascular system development first. And in the first episode of development of cardiovascular system, let us talk about formation of the heart tube and its bending to form the cardiac loop. The heart, which is center of the cardiovascular system and the first organ to start functioning in the intrauterine life, starts beating as early as day 22. And by about day 25 or 26, it actually starts pumping blood. The heart forms as an endothelial tube that folds and get partitioned as a result of series of processes like proliferation, migration, programmed cell death and remodeling to eventually culminate into a definitive four-chambered masterpiece of a pump that we all know as the heart. Let's just see these events from the beginning. The vascular system in an embryo appears in the third week. Till now, from second week till third week, the uteroplacental circulation that was established at the time of implantation was somehow managing the nutritional needs of a rapidly growing conceptus. But from now on, the uteroplacental circulation probably is not going to be enough. So the heart starts developing. The cardiac progenitor cells migrate from the primitive streak to splanchnopluric mesoderm, cranial to the buccopharyngeal membrane, and they appear to concentrate along a horseshoe-shaped area known as the cardiogenic area or cardiogenic plate, or simply speaking, heart-forming plate. This horseshoe-shaped area is the primary heart field. But mind you, primary heart field is not the only area that will contribute in formation of the tissues of the heart, because it does not simply contain enough amount of those cardiac progenitor cells. So there is establishment of a secondary heart field area as well. And this secondary heart field area is located in the splanchnic mesoderm ventral to the pharynx. In this cardiogenic area, which is at the splanchnopluric mesoderm that forms the floor of the intraembryonic coelom, is located cranial to the procaudal plate. In the midline, cranial to the buccopharyngeal membrane, the intraembryonic coelom of both sides gets continuous and the pericardial cavity is formed from this very part of the coelom. Cranial to the cardiogenic area, the somatopluric and splanchnopluric mesoderm are continuous, and this unsplit part of the mesoderm is called as septum transversum. This septum transversum later gives rise to a part of the diaphragm along with other derivatives. The splanchnic mesenchymal cells in the cardiogenic area aggregate and arrange themselves bilaterally as angioblastic cords. The cords canalize to form two endothelial heart tubes. With the lateral folding of the embryo, the heart tubes approach each other and fuse cephalocaudally to form a single tube. Just to get more clear idea about the relative positions of the developing heart tube, the pericardial cavity and other structures in the embryonic body, before folding of the embryo, the developing heart lies cranial to the procaudal plate, caudal to the septum transversum and ventral to the pericardial cavity. In terms of pericardial cavity, it can be said the pericardial cavity lies cranial to the procaudal plate and caudal to the septum transversum. With the cephalocaudal folding, the heart tube comes to lie ventral to the foregut, dorsal to the pericardial cavity and cranial to the septum transversum. From the pericardial cavity point of view, the pericardial cavity lies ventral to the heart tube and foregut and cranial to the septum transversum. Now the heart tube invaginates into the pericardial cavity and is surrounded by a double-layered pericardium filled with pericardial fluid. So visceral layer of splanchnopluric mesoderm will be in contact or closer to the heart tube, and parietal layer is the somatopleuric mesoderm. The heart tube thus formed remains suspended from the dorsal wall of the pericardial cavity by a fold of mesodermal tissue called the dorsal mesocardium. As the heart tube is functioning, as the heart tube is gaining length, the central part of the dorsal mesocardium soon shows opening, or it degenerates and gives us to a rent, and the connection thus is established between the two sides of the pericardial cavity, and this connection is the one which forms the transverse pericardial sinus. And now the heart is suspended in the pericardial cavity by its cranial and caudal ends, and now the heart is there to show a series of dilatations and constrictions. The three constrictions appearing are namely sinoatrial groove, atrioventricular groove, and bulboventricular sulcus, as a result of which it shows four dilatations. They are sinus venosus, atrium, ventricle, and bulbus cordis, where sinus venosus represents the venous end of the heart tube. Bulbus cordis, on the other hand, represents the arterial end of the heart tube. The sinus venosus is separated from atrium by the sinoatrial groove. The atrium is separated from the ventricle by the atrioventricular groove, and the ventricle is separated from the bulbus cordis by the bulboventricular sulcus or groove. The ventricle and atrium are connected by a narrow canal called atrioventricular canal. The sinus venosus is connecting with the atrium at the sinoatrial groove, as we have already seen. This sinus venosus shows two prolongations in the form of right horn and left horn. The bulbus cordis lies at the arterial end of the tube, and it is subdivided into three parts: a proximal 1/3, a middle 1/3 called the conus cordis, and a distal 1/3 called the truncus arteriosus. The truncus arteriosus is continuous cranially with the aortic sac. The aortic sac is continuous with pharyngeal arch arteries, and the arteries become continuous with the dorsal aortae, and there are two dorsal aortae which will supply blood to the different developing parts of the embryonic body. This heart tube with four dilatations is now ready to show some more growth, and as a result of which it is going to bend. So let us see the looping of the heart tube. What are the factors responsible? Out of the many factors implicated, the first one is constant growth of the heart tube by addition of cells at both the ends. The other one is biomechanical forces, where flow of blood through the heart tube and cardiac contractions make certain biomechanical forces available in the surrounding area which stimulate the heart tube to bend. The third factor would be tension. Dorsal mesocardium is thought to play some role in bending process by providing some tension due to suspension of the heart tube. Even the signaling molecules are also implicated in that. Signaling pathways involving genes like LEFT and PITX2 are involved in establishing direction of the loop, and the other factors are convergent extension and unequal growth. With so many factors operating at the same time, on day 23, the heart tube begins to elongate and bend. The cephalic portion of the heart tube, the one which is closer towards the arterial end, bends ventrally, caudally, and to the right. The caudal portion shifts as a result dorsally, cranially, and to the left. It means the bulbus cordis is going to move towards front, caudally, and to the right, and the sinus venosus end of the heart tube is going to shift dorsally, cranially, and to the left. This happens through two stages. At first, there is deepening of the bulboventricular groove, making the heart C-shape, and later on, with cranialward migration of the sinus venosus, this C-shaped loop of heart tube assumes a S-shape. The cardiac looping is completed by day 28. As a result of this cardiac looping, the four chambers come to occupy their respective positions. The sinus venosus now opens into the primitive atrium from the dorsal aspect, and the truncus arteriosus gives attachment to a series of arterial arches which will be connecting to the dorsal aorta. This prepares the foundation for further development of the different parts of the heart, like the atria, where they are going to segment into two chambers. The ventricles will also experience septum formation to form right and left ventricles, and the outflow tract for both the ventricles are to be formed by formation of yet another spiral septum. So all that will follow in the next upcoming episodes. Happy learning.